I'll be talking about artificial cells. So the objective in the research in my group is to build a cell in the lab, and I'm just going to go very quickly through what we're really trying to do. So as you know, this is as you can see, this is a Lego block, and I'm sure you're not playing anymore with this, but maybe your kids are. And you can make lots of different objects from these components. And you can also make this kind of things. But now, with the advance of technology, we biologists or biochemists can start working with molecules, nucleic acids, proteins, and phospholipids, and starting from only using these defined components to build cell-like structures. And so this is the schematic of our artificial cells, where its size is around 10-20 microns and encapsulated by phospholipid vesicle, and the central dogma, meaning the DNA, mRNA, and protein reaction takes place inside. So it really mimics kind of the real cell in some aspect. And so I just show you our favorite, actually not our, but my favorite movie of our synthetic artificial cell. Just very briefly speaking, this artificial cell, the membrane is stained with orange, so that's why you see some edge in this circle, and inside it's stained with green and red, and the green molecule is pretty small. And what I'm showing here is that in the beginning, because the green and red is overlapping, all the artificial cells are yellow, but now it turns red afterwards, meaning that these green molecules get secreted outside from our synthetic cell without using membrane proteins. And I don't have time to explain the details about it, but it's really a very simple chemical system, but still can show some properties of a real cell where the materials inside can be transported outside, and I'm not showing here, but there's also a way to incorporate chemicals from outside to inside and encapsulate this. And so this kind of features can be reproduced with a very simple setup, although the current living system is using hundreds and thousands of molecules to do this kind of things. And so why we're doing this kind of artificial cell synthesis research, I would like to explain in this slide. And of course, I'm not the only one, or I'm not the first one to say that this kind of approach is important. I just refer one paper from Shostak already 20 years ago, where he explained the importance of synthesizing cell in the lab. So as many of you know, if you're not a biologist, you don't have to know everything, but I mean, the current cell is really, really complicated, and it consists of hundreds and thousands of different molecules. But of course, it shouldn't be like this from the beginning, and it must have started much simpler form, like some methanes or whatever, phosphates and something, and gradually through chemical evolution have become more and more complex, and at some point had become like so-called a protocell, and through the course of evolution turned out to become like a modern cell. This is one possible scenario. I'm not saying that this is the answer. Nobody knows what the answer is, but the important point is that it's very likely that it started from simple chemical and in some way ended up in a more complex system. So this part is kind of prebiotic chemistry, and what we're doing in my group is to construct a protocell, because the prebiotic chemistry shows like how amino acids can be made, how nucleic acids can be synthesized, and so on and so forth. But I mean, amino acids, nucleotides, yes, these are the elements or the components of the living system, but does not necessarily mean that life is there. Life is a system. It's not a molecule where multiple molecules are working together as a molecular system. So we tried to start from biomolecules and try to assemble living systems, And the challenge is we try to do this, and we aim to create artificial cells only from defined component. And the objective of this research is, first of all, to understand how biological system might have emerged. So it's kind of origins of life research. So if we are, or if I could, or if my group member can make the real cell in the lab, that's one possible pathway. So I'm not sure whether that's a really historical event, but it also means that that's one strategy to build a cell, and that's a very big achievement. And the other objective is how biological system can be assembled or synthesized itself is also a very challenging thing. So as a bioengineer or synthetic biologist, we really want to make a system which behaves like a cell, but it's still, right now, nobody has ever been able to do this. So technically, it's very challenging, and if we can do this, we can also learn about origins of life. And so that's why these are the two main objectives of why we're trying to do this research. Now, going back to what I called artificial cell, I would just want to go a bit more in detail about what I mean by artificial cell, a bit more in detail using the materials. So again, starting from molecules, nucleic acids, proteins, and lipids, we make this kind of things. And here we have a system called Pure System, which is an in vitro translation system which makes a protein from DNA. So the whole central dogma is incorporated in this compartment, micrometer compartment. And this is the result when we encapsulate GFP encoding DNA. GFP is encoding a green fluorescent proteins. So you can see this is the size is like 5 microns, and you can you can see that the green fluorescence is inside, indicating that the protein synthesis is occurring inside the vesicles or the artificial cell. So, and we can also analyze the time course of protein synthesis, time versus GFP concentration, and you clearly see the increase. And very interestingly, this reaction can be done in bulk, meaning in microliter scale, and also can be done in this artificial cell, or in other words, we can call this liposome, and the size is femtoliter. So the difference is like 10 to the power of 9-fold difference, but you can see that the time course looks pretty much the same. So we're able to really make this kind of artificial cell that can really do the complex reaction in a really confined environment. But now the point is that this synthetic cell or artificial cell just produces proteins in this way. So there's just a monotonous increase in the protein concentration. However, many of you know that the real living system is very different, and it's really highly regulated. And so I'll just take some time to introduce how the current living system is regulating or responding to environmental changes. So this is the gene encoding gene Y, and here's the X binding site, and X is actually an activator, and I will explain to you what this whole thing is. So if molecule X binds with something called SX, which is an environmental factor, it can be hormone or it can be some small chemicals, whatever, it changes to X star, and this X star then binds to this upstream region where it's called the activate X binding site. And having this molecule here, the RNA polymerase will get recruited to the DNA, and the RNA and the protein will be synthesized. So this is how the living system regulates the protein expression. So to summarize this in a plot, this is time versus concentration of protein Y. You can see that, for example, in the case of environmental factor, for example, SX, is now 0, but there's a spike like this, like this, like this. And now how the system responds is that here, the translation machinery gets switched on and off and on and off, depending on the environmental factor. So this is a very simple form, But as I showed you in the previous slide, what we can do, or what the community can do, is that, okay, we incorporate the protein translation system. Now you can synthesize proteins, but it's very difficult to make this kind of regulatory system. And so it's not easy at all for an artificial cell to respond to environmental oscillation and switch on and off the protein expression as in the living system. So today, I want to talk about two challenges which we are working on recently. And that is to construct an artificial cell that responds to environmental oscillation. So this is time versus environmental factor. It can be anything, but the condition is changing up and down. It can be the concentration, the concentration of chemical X, or it can also be pH. It can be light, or whatever. And so first of all, the question we wanted to address here is, how can we make the artificial cells to respond to chemical environmental oscillation? This is already a very big challenge. As I showed you before, it just monotonously increases, and how we can design a system which really looks like a real cell is a very big challenge. And so the second question which I would like to address in this talk is, now, how do this kind of environmental oscillation affect the artificial assembly? And this is something related to origins of life. And so, of course, at the completely equilibrium condition, the molecule just stays as it is, and it's just dead. And but by having this kind of oscillation, it could be temperature, it could be light, pH, or something. This kind of energy can be used to assemble or create a living system. And so I want to show you these two examples. So more precisely speaking, in the first part, we show that the artificial cells that switch protein expression depending on the chemical oscillation. So I will show you that indeed our system can respond to environmental oscillation. And second, we will show that the artificial cells can be assembled using one of the environmental oscillation, namely the freeze-thaw cycle. And we'll start from this engineering part. So as I told you, our current artificial cell can make protein, looks pretty small and looks like a cell, but cannot respond to environmental oscillation. And so in order to achieve this, we make this completely synthetic. So the mechanism or the machinery we use is called the riboswitch, but I will explain to you a little bit about what I'm going to be talking about from now. So this, since, I don't know, 30, 40 years, RNA has been very well recognized as a functional molecule. And for instance, there's a naturally occurring guanine binding RNA. So this particular RNA binds to this guanine. And since the discovery of this kind of naturally functional molecules, maybe you know very well, that researchers have started constructing or discovering a variety of RNAs that has multiple functions. And for example, this paper, it's already 5 years ago, this was the work together with Yokobayashi-san from OIST. We discovered an RNA that binds to histamine. And recently, my group member Fukunaga-san found an RNA that binds to some drug called ASP2905. And we also found proteins, RNAs that binds to particular proteins, and so on and so forth. So the message is that, okay, RNA can be modified in a way that it recognizes a particular molecule of interest. And so these kind of RNA will be used in the next few slides to install a new function, namely in a function to change, to respond to the environmental changes. And so I will very briefly explain how this aptamer or the RNA sequence can be used to regulate the fate of the artificial cell. So the DNA construct looks like this, where there's a DNA promoter, and here is the histamine aptamer encoded, and a ribosome binding site, and M-cherry is just a red fluorescent protein, which is used as a marker. And the important point here is that once this DNA is transcribed into RNA, it makes a secondary structure like this, where the ribosome binding site will be hidden or bound by this particular RNA, and therefore the ribosome, which is the machinery to translate the protein, do not have access to messenger RNA, and therefore this protein cannot be synthesized. However, once the histamine comes, the RNA binds, because this aptamer binds to this histamine, this ribosome binding site gets open, and the ribosome was off, but now it switches on, and the downstream messenger M-cherry protein get expressed. So we can test this, the ability of this DNA using a cell-free translation system. It's not very, the name is not very important, but it's a system where you can synthesize protein in a test tube. So what I'm showing here is the time versus M-cherry fluorescence, namely the protein expression of this downstream protein. With histamine, you get a bunch of proteins, and without histamine, there's almost nothing. And so we could really regulate the expression of the downstream M-cherry by the presence of histamine in this way. And the very important thing is that this does not exist in nature. It was made completely synthetic, so it's completely artificial. So what we can do more, we can have an artificial cell, and upon the addition of histamine, we can have a system where the M-cherry protein can be synthesized, or if we encode another protein, so we can make a synthetic cell, artificial cell, that makes a pore, which allows the leakage of only the small molecules while all the large molecules are maintained. Or we can also encode a protein that will induce the kill switch, so it's kind of a committed suicide, or like apoptosis, if you want to say. And so to just show you one example, here is our artificial cell, which then responds to histamine, and in this case, we encoded an enzyme called phospholipase, and the name isn't important, but upon the addition of histamine, this artificial cell just dies. And if you look at the confocal microscopy image, so these are the calcine and ovalbumin, that doesn't matter, but these are the artificial cells, the size is around 5 microns, but upon histamine, you see the fragment of a membrane, and in this way. So this kind of completely synthetic response can be synthesized or constructed in a bottom-up way in the lab. So the take-home message here is that we created first the new riboswitch. So the response element is new, and the output, meaning the killing suicide switch, is also synthetic. So whatever input can be converted to whatever output you want. And so this kind of design can be applied with our artificial cell. And so that's a very big advantage of this kind of system. So we can make the system more and more complex, and finally one will make it respond to the environmental changes, namely the chemical concentration. So let's start with developing another riboswitch sequence, which then first has the ribosome binding site hidden, but upon the binding to some chemicals called ASP something something, the ribosome binding site gets open and the downstream GFP sequence get expressed. And this is another one which responds to theophylline. And these two were developed very recently. And I show you the result here, where you have this ASP2905 riboswitch encoded upstream of GFP. And if you don't add anything, of course there's no GFP fluorescence, but if you add ASP, you see a very strong fluorescence, But if you add theophylline, there's almost nothing. And for this one, you see the theophylline switch is switched on, but which it does not respond to ASP. So in this way, we're now able to control the expression of multiple genes simultaneously by adding a synthetic molecules. And so by having this tool, we now can make the artificial cell to respond to chemical environmental oscillation. So what I mean by this is that, for instance, in case we have ASP2905 riboswitch here, we add this chemical. Okay, this is time versus GFP fluorescence. And if you just have ASP2905 all the time, you see a monotonous increase. So it's just like as I showed you before. But here we switched on and off. Off means we remove this ASP2905 from outside, and then the protein expression stops. And if we add it again, it switches on once again. And we can do this with theophylline as well. Switch on, off, and on. And in this way, so this is white, gray, white, so the environmental is changing, and the protein expression is responding indeed to this kind of chemical oscillation. And so this kind of things can be constructed in this way. And we can play around a lot more with it, make more and more complex. So we can incorporate two genes simultaneously, where it can respond to both ASP something and theophylline, and switch on and off. So switch on the one switch for GFP expression, and the off for this red fluorescent proteins, and so on and so forth. But I mean, the main message here is that now we were able to construct an artificial cell that responds to multiple environmental oscillation. And so this kind of things can be done completely synthetic. And we now developed more and more synthetic riboswitches to allow the artificial cells to do more and more complex things. So it might to you sounds like a complete engineering, and it is indeed an engineering, but in my opinion, first of all, for human being, it's still not possible to make a real living cell. So this kind of stepwise increase in complexity, at some point, we believe that we'll reach, at some point, we can, to some kind of a system which we can call a living cell. So now going to the second topic about how to use environmental oscillation, or how do environmental oscillation affect the artificial cell assembly. So far, I just explained to you about how to construct an artificial cell that responds to environment. But now I'm going to be talking about how this kind of environmental oscillation can be used as an energy source to build artificial cell. And so, as I have been showing many times, so the cell must have started from simple to complex, but we aim to reveal how biological system can emerge under a bit more geologically pulsable oscillation condition. And of course, in the previous slide, I used lots of things like, a plate reader, ultracentrifuge, blah, blah, blah. These high-tech machines are used. But of course, during the course of evolution, of course, there's no ultracentrifuge or something like that. So whether some kind of geologically pulsable condition can lead to, for example, construction or the development, creation of a synthetic cell or artificial cell is kind of a challenge we wanted to do. And as an environmental changes, I don't think I have to explain anything about this, but we used a freeze-thaw cycle because this is present and ubiquitous, of course, on Earth, but also present outside Earth, very surprisingly. And I learned this from Sekine-san, and I was really shocked, and I didn't know that this kind of thing exists. So I thought this would be really interesting to see, because freeze-thaw condition is present not only on Earth, but it also exists outside. So if is it possible that this kind of freeze-thaw conditions can be used to assemble biomolecules or living systems. So again, using freeze-thaw is a very attractive approach, but I'm not actually the first one to think about this. The group in UK, namely Philip Holliger's group, have used the freeze-thaw cycle to accelerate the ribozyme reactions and so on. But the key point here is that if you freeze the liquid very slowly, there's a phase called eutectic phase will appear. And in this narrowed channel, all the solutes like ions and molecules get concentrated. And this kind of behavior was used to accelerate the ribozyme activity in a confined space. And this was already published like 10 years ago. And the other group, again, this is a group from Germany. They have shown that the freeze-thaw cycle can use to proliferate the self-replicating ribozyme inside vesicles and so on and so forth. But now we wanted to ask, can freeze-thaw cycle do more to this? And to go back to what we really want to do is that we, again, we assume that the biomolecules are there. We're not doing prebiotic chemistry. We skip the whole part. We think that, okay, in some way or another, the biomolecules has been emerged through chemical evolution, but that's not life. I mean, that's just amino acids, nucleic acids. They have to be converted into a system, not like this, but in some way this way. So how can this be achieved is the question we wanted to address, and more specifically, we wanted to ask how freeze-thaw cycle can contribute in this process. So the first thing we did is we started putting biological material, first of all DNA into freeze-thaw cycle, and then a vesicle into freeze-thaw cycle, and ask what will happen to these kind of molecules. And because the reason why I'm showing here is because we found that the freeze-thaw cycle does very interesting work. So what we did here is that we prepared two very long DNA fragment, which is 2 kilobase and 3 kilobase. But the trick is that it has a 50 base pair, a base complementary sticky ends. So it has a very long sticky end, and these two can hybridize each other. However, because the size of these molecules are very, very large, even if you mix these two together and just wait for a while, none of the 5 kilobase band appears. However, if you subject this sample into freeze-thaw cycle, we start seeing a band up here. So it means that this product is appearing only when it's subjected to freeze-thaw. So now what do I mean by freeze-thaw? This is time versus temperature. What we did, freeze the sample to -30, let it stand, thaw very slowly, and put it back. And these guys, we just let it stand at 25 degrees for the same period of time, but we don't see anything. And so it was a very, very promising result in the beginning. And so we did a systematic study. This is the DNA concentration versus the hybridization efficiency. So if everything gets hybridized, it's 100%. But the important point is that regardless of the DNA concentration, at very, very low concentration, low nanomolar concentration, this is the hybridization efficiency with a freeze-thaw cycle, all remains around 20%, whereas if you don't do freeze-thaw, it's almost nothing. It's like few percent. So very strong hybridization acceleration was observed. And we also checked the magnesium ion concentration dependency and so on and so forth. And the conclusion is that both DNA and magnesium ions were concentrated, very likely because of this eutectic phase. And so we can go more, because I told you slow freeze and thaw is important. But where is the more important part can be done systematically, because we're doing everything in the lab. And so we have a bunch of data here, but I just want you to, because the conclusions are the same, I just want you to focus on one particular result. So I have 10 minutes left, 5 minutes left. 5 left, okay, so I'll go a little bit faster. And the point is, to make the story a bit short, the slow free thawing was very, very important. And so I don't have time to explain in detail, but having this material between -10 to 5 for a long period of time was very important for this high efficiency, hybridization efficiency. Now going back to the environmental oscillation, we can repeat this 16 times, freeze-thaw, many, many times. And what you see here, this is hybridization efficiency. It was like 5 to 10% in the beginning, but if you repeat it multiple times, you get more and more longer DNA. And after like 16 hours, it reaches like 40%. Now we can work with much longer DNA, and the conclusion is the same. The more the DNA experiences the oscillation, freeze-thaw cycle, the more the DNA gets hybridized. So what actually we want to do right now is that we want to construct a minimum genome. So this is the plot of the DNA genome size versus the number of gene, and it's known that the minimum gene is around 500 kb. And if we can start from prebiotically possible like short oligonucleotides and subjecting this into freeze-thaw cycle to reach a 500 kilobase DNA, we now reach 20K. So we now can go like 25-fold higher. Then we can say that, hey, man, we're close to making life, at least in respect to DNA length. And now going to the second topic about putting the lipids into a freeze-thaw cycle. So what we did, we prepared 3 different vesicles, and I marked with different color, and this color indicates the difference in chemical composition. And the difference is super, super small, so I just mark it like this. It's called POPC, PLPC. Please remember only the name. It's called POPC and PLPC. It has 2 acyl chains and head groups. And the difference between these two is the presence of 1 unsaturated bond. That's it. It's really tiny, small. And we prepared vesicles with POPC, PLPC, and mixture, and investigated what happens if we put these vesicles into freeze-thaw cycle. And I'll show you the result with DLS. It's measuring the size of the particle. So 100% to 100% PO to PL. In the beginning, we started all from 100 nanometer vesicle, and when we subjected this to freeze-thaw, we start seeing a huge particle, especially in those with PLPC, and indicating that this PLPC vesicle is growing much more than this POPC vesicle. And we can quantify the amount of this large vesicles, and we found that around 80% of the vesicles grow from 100 nano to few microns. And important message here is that this is very strongly dependent on the chemical structure, namely PLPC works the best. Now, that was how it grows, and I'm not going to go into detail about this data, but we do a lot of quantification and asked how well the membrane is mixed. And I just show you the final result. This is freeze-thaw cycle versus how well the membrane was mixed, and the conclusion is that the PLPC was the best again, as in the first slide. Now, we also tested how well the contents can be mixed, and the conclusion, again, is the same. PLPC is the best. So we compared how well the vesicle grows, how well the membrane mix, how well the contents will be mixed. Everything was best for PLPC. And now we wanted to do, why we wanted to do this is because we wanted to understand what happens if we put these samples into a freeze-thaw cycle, for example, in Europa or something. So what we did here is that we prepared blue vesicle and yellow vesicle, and blue is PLPC and POPC, and subjected this to freeze-thaw cycle. So in the beginning, we have diversity. We have yellow and blue coexisting together. We put it into freeze-thaw cycle and ask, what are the vesicles which grew? And what we found is that PLPC, namely the blue ones, get enriched from one-to-one mixture to like twofold by, this was quantified by HPLC. So what I'm trying to say is that very simple trick, freeze-thaw, enable the chemical selection of a particular lipid in this way. But we want it to be a bit more prebiotic, so we now incorporated DNA inside. So the name of the DNA is not important. So the PLPC has red DNA, and PLPC had green DNA. And what we expected is that, okay, this red DNA also wants to get transmitted to the next generation. And actually, that was exactly what happened. And so the red DNA survived more in the grown vesicles. So if this kind of vesicle population is present in aqueous solution, and if this goes through multiple rounds of freeze-thaw cycle, what will happen is that specific DNA enrichment occurred simultaneously with the enrichment of the compartment, namely the phospholipids. So this kind of things can be seen with a very simple setup, namely the freeze-thaw cycle. So this is the last slide, the summary. By putting the biological materials like DNA, vesicles into freeze-thaw cycle, we showed that the DNA can get longer with freeze-thaw cycle, and simultaneous chemical selection of compartment and DNA can occur. And so, again, the question we want to address is how can we make pieces into a system. So we now showed this kind of thing. So we would now start making the system more and more complex and ask how far we can go or how close we can go to an evolvable protocell. And of course, freeze-thaw may not be sufficient. In this case, we might need another environmental factors to really fully achieve this kind of things. And I talked as I did the research, but I didn't do anything. I'm just presenting here as a representative of the lab, and these are the people who really did the work, and I really appreciate these people and the funding, and thank you for your attention. That's all for me.